you still remember biology class and know what a gene exactly is? If not, I'll explain it to you very simply in this video. Let's start with the human body. As you know, we are made up of many trillions of individual cells, and in almost every one of these cells is the cell nucleus. If we take a closer look at the nucleus, we find the chromosomes, which are these X-shapes you probably know from television. A chromosome, in turn, consists of a tightly coiled thread, the so-called DNA double helix. That's the twisted ladder you probably already know. This DNA contains the genetic code, which consists of only 4 different genetic letters: G, A, C, and T. In the entire cell, there are around 3.2 billion such genetic letters, which are present in several long chains in the cell nucleus. And now we come to the genes. Genes are specific regions in this genetic code, but they contain something essential: instructions for the body to perform a specific function. To humans, a gene looks like a jumble of letters, but the cell can read these instructions and make something useful out of it. For example, there are pigment genes that contain instructions for producing a brown pigment. This then becomes visible in brown eyes. Alternatively, there are blue and green variants and give each of us a different eye color. This is all greatly simplified, but the principle remains valid. A gene tells the body how to perform one specific function. Since most genes control only a single function, we need many different genes to enable the complexity of our bodies. In total, we humans have around 23,000 such genes, each performing different tasks. A good example of a gene is a Lego construction set. I will now play the role of the cell and take a closer look at one of these 23,000 genes. Let's take the LDLR gene, which is responsible for cholesterol regulation in the blood. So as a cell, I decide that it's time to listen to this gene, and the gene becomes active. We can imagine the gene itself as a construction manual for this excavator. So the gene contains the step-by-step instructions of how to assemble this structure. The first step is done. I know what I need to do, but now I'm missing the building blocks. But that's not a problem, because these building blocks are floating around everywhere in the cell, just waiting to be used. There are only 20 different types, the so-called amino acids. We get these from food or produce them ourselves in the cell. Just like with Lego bricks, there are amino acids that are particularly good at holding together. Then there are others that have a supportive function, and still others that are good at grasping certain things. I follow the gene's construction instructions and assemble one amino acid after another until we have the finished excavator in the end. In biology, we call this construct a protein. And as you can see, the way we assemble the building blocks gives this protein a specific function. This protein is obviously good at digging. Since we replicated the LDLR gene, this specific protein sits on the surface of the cells and catches passing cholesterol with its shovel. This filters the cholesterol out of the blood, transports it into the cell, and lowers cholesterol levels. Good news for the heart. This is what the LDLR excavator looks like when it's catching cholesterol in real life. The cell is packed with various proteins. Some are support structures, others work together to copy DNA, and others break down nutrients, and so on. Now we come to gene defects. These instructions are complex, and as you remember, the same construction manual is in each one of the 50 trillion cells. So the cells must be really good at copying these instructions error-free. Sometimes a copying error occurs, but most of the defects in our genes are inherited from our parents. What happens in the case of a gene defect? In most cases, a wrong amino acid is used for construction. If it affects an essential part of the function, we might build an excavator without a shovel. The cell doesn't know about this and diligently produces all of these useless excavators for the cell surface. Naturally, the cholesterol can no longer be captured. The result? High cholesterol levels and a sick heart. With modern technologies, we can now read these construction manuals of humans and search for errors. For excavators without shovels, we can then very reliably say the cholesterol will not be lowered properly. Today, we cannot correct these errors, so we have no choice but to look for other ways, such as diet, to lower cholesterol levels through the diagnosis of the problem. And that's the basic principle of preventive genetics. We identify genetic weaknesses and try to counteract them in other ways.